Hello everybody, welcome to the Rahbar Talk show. Today we want to perform our second session related to calorie restriction and their possible effect on eukaryotic system. How the calorie restriction can modify cell bioactivity and which signaling pathway are involved in the promotion of the cell's bioactivity. Let's see what happened here. Dear Daniel, as you remember, you told us about the possible effect of calorie restriction on body fats. Maybe the calorie restriction and make some lipolysis. Can you make any additional information about how this correlation exists?
Yes, Professor. As you mentioned in the previous session, I discussed about the weight loss aspect of fasting, intermittent fasting, and calorie restriction on our body. First thing we should know is that the most immediate organ which is affected by fasting is pancreas. During low plasma glucose, pancreas will release glucagon from alpha cells. Glucagon will mainly affect liver as it stores the most of the body's glycogen. After hepatic glycogen stores are depleted, body starts using adipose tissue and protein for energy. Meanwhile, it should be pointed out that the liver is the main organ for metabolism of fats, given that it's the main oxidizer of triglycerides. Fasting is accompanied by the gradual emptying of fat stores, resulting in the release of fatty acids and glycerol into the bloodstream and the production of ketone bodies in the liver. Adipose tissue is the most important energy storage in our body and has critical importance during fasting. Energy is deposited in adipose tissue as triacylglycerols. Adipose tissue consists primarily of adipocytes, which contain a large central vacuole filled with triacylglycerols.
Sorry, that the adipocytes you mentioned.
Yes,
great.
Which is filled with triacylglycerols and cholesterol esters.
So we have some million numbers of the free fatty acids. They are directed to the hepatic tissue for just anabolic steps.
That's true. Also, moreover, adipocytes constantly collect, store, and break down triacylglycerols. And the turnover rate of triacylglycerols for an average size adult is 50 to 100 grams. And it's worth mentioning that adipocytes are, given that they store most of the body's energy, they are central for metabolic response of our body to fasting. Several metabolic mechanisms are activated and repressed during fasting.
Let's just elucidate what happened inside the hepatic tissue. Upon reaching to hepatic tissue, this free fatty acid is going to conjugate with other molecules like HDL, LDL, and so they are just transferred to the peripheral tissue. So the other cells are located in different tissue and different microenvironment can use this fatty acids.
That's true. That's completely true.
So, any comments? Elskom?
Several metabolic processes are activated or repressed during fasting. For example, in adipocytes, For example, fasting stimulates the breakdown of triacylglycerols, and as a result, releasing non-esterified fatty acids and fat stores into blood. And on the other hand, fasting represses the circulating triacylglycerols and results in a reduction of uptake and storage of fats in adipocytes.
But the other cells can just absorb the free fatty acids from the circulation.
Yes, dear professor, that's true. And if you let me, I'm going to give more elaborate in the next round.
Okay, thanks a lot. It's nice to see you. Thanks a lot. Mr. Khostabi, as remember, you talk about the activation of the some molecular pathways upon the calorie restriction inside the cells. And you just made some brief communication about the insulin signaling pathway, as I remember.
Yeah, of course. As I mentioned previously, in nutrient deprivation, there are three factors that play role. Energy sensors, signaling pathways, and transcription factors. Energy sensors like ATP, when we are in a calorie restriction situation, ATP level is low in our body, and that promotes AMPK, that reduces AMPK function. And in this situation, fat burning and ketone bodies production will be high. mTOR in normal condition promotes cell growth and cell proliferation. When AMPK is low, mTOR will be turned off. And
we have two kinds of the mTOR subset, the mTOR1 and mTOR2. So the upon activation of the AMP kinase, the mTOR1 is switch off, is deactivated, So maybe you are talking about the mTOR2, I don't know.
mTOR2, that will promote autophagy.
This is a type 1 mTOR. It's switched off, but simultaneously autophagy apparatus is activated.
Yes. On the other hand, in this nutrient deprivation situation, some signaling pathways will change. Like when insulin and IGF-11 in our body is low, PI3K and ACT pathways function will reduce, and that will activate cellular stress resistance, helps cells to be healthy, energetic.
Mr. Yalame, as your friends mentioned and highlighted, insulin signaling pathway is one of the target of calorie restriction. How do you think that this signaling pathway can just regulate cell bioactivity under bioenergetic stress?
Calorie restriction or CR can suppress the releasing growth hormone and then insulin IGF-1 signaling pathway. That leads to the transcription factor FOXO3 translocate in nucleus and upgrade the genes involving DNA repair, stress resistance, or cell cycle arrest, and autophagy. I think this pathway is strongly related to increased lifespan.
You mentioned that upon the bioenergetic stress, some signaling pathways are activated, which coincide with the downregulation of the some signaling pathway. For example, insulin signaling pathway is deactivated upon what? Upon what? The reduction of the glucose, the systemic glucose. So it's going to just the inhibition or stimulation of some kinds of the gene and effector inside the cells, and this enables the cells to just, and potentiate the cells to just restore its function by just, making some replacement of the injured subcellular compartment. Yeah, am I right?
Yes.
Great, thank you a lot. Thanks a lot. Mr. Nazif Kerdar, the autophagy response is one of the most interesting targets during the calorie restriction. And so the cells which are under biogenetic stress have higher autophagic activity compared to the healthy cells, or even that the cells are under normal conditions. So what is autophagy?
So, as we earlier mentioned and aforementioned in our last sessions about the autophagy, I would like to briefly have an introduction to the autophagy, as we mentioned here.
Right.
Autophagy names that when the cells are under stress, maybe our cells are going to die and some organelles are going to die and some proteins are going to be misfolded in our body. So autophagy activated is going to activate it in order to clean these kind of things and get rid
Which
of
kind of things, sorry?
organelles, misfolded proteins, and cell debris, dead ones. They're going to get rid of the clean out from the body by the autophagy mechanisms. And also autophagy mechanisms, recent articles shows that there are three main types: macroautophagy, microautophagy, and chaperone-1 mediated autophagy. And the most important one here is the macroautophagy. And it is in the structural aspects, it is going to initiate by the producing phagosomes. And in later steps, these phagosomes are going to be cell double membrane vesicles, is going to fuse with the lysosomes and surrounded the cells and secreted some acidic hydrolysis enzymes in order to enzymatically degrade those cell debris or dead ones and organelles that are going to cause some kind of inflammation, maybe some kind of cancers may be going to happen in our body, and other pathological illness.
Yeah, you are right, exactly right. Under some stressful condition, the ability cells to just exclude or eliminate some of the abnormal molecules can help the parent cells to just away from the stressful condition. It's a good strategy. As you know, the autophagic response is activated during the 1 to 6 hours after energy deprivation. And so this is one of the early stage phenomena that will happen in cells, in different kinds of cells, which are directly exposed to some stressful condition. Thanks a lot.
And another comment, may I add? This is when we are under the autophagy phase. Usually, cells would like to be in proliferation phase in order to get produce lots of cells and get
which kinds of cells? As you know, the cell with autophagic
cells in the body usually would like to be in proliferation.
But this is after this will happen after autophagic response.
And when the autophagy is going to activate it, our body is on the repair mood. Our body is in repair mood and maintains mood rather than proliferation.
Yeah, absolutely. It just helps the immune cells component and even the other cells to just be resistant to and just repair and replace their parts. Even that, but it should be just regulated. As you know, the excessive autophagic compared to the adaptive autophagy, as you mentioned, is detrimental. Thanks a lot. Farzin, as I know, as mentioned in different research and studies, there are different kinds of the effectors which are involved in autophagy machinery. They can just orchestrate this process during the different physiological and pathological condition.
Yeah, actually, there are lots of genes and proteins that activate during autophagy, but there are most of important ones are ATG genes like 1, 2, 5 and ATG16L1, but there are some another important genes that usually activates and deactivates or converts to other types. Actually, the key factor that activates autophagy is Beclin-1. I mean, if the expression increases, it means that autophagy is going to start.
It's also initiators of the autophagy.
Yeah, it initiates the autophagy. Also, if you look from another point of view, the ratio of conversion of LC31 to LC32 is really important because P62 also activates, which is really important, and it combines the misfolded proteins, damaged cells, genes and proteins which are destroyed, and it works like a shuttle that combines them together, and it helps the LC32 to absorb them and elongates the autophagosome. And if autophagosome elongates, it means that it's activated and the autophagy goes on. So it's really important to understand the dysfunction and these genes expression, down-regulation, up-regulation, which is really important.
So LC3 is a kind of factor just help the autophagism to just direct it toward lysosomes like in collaboration with P62. And also the LC3 is kind of the microtubules protein. It acts as an anchor to just fixed and stable the autophagosomes, and then they are directed toward lysosomal degradation. It's great. But as you know, the overactivity of the autophagia is not a good. Can you make any, brief communication about this?
Yeah, actually, in most of the papers that have been published, it's mentioned that autophagy is like a two-way sword. It means it depends on the age, on the situation of the person, on the stage of the illness or the cancer which is going to happen in the body. I mean, in the first stage, it's survival. Autophagy helps the normal cell to stay there, to survive, and also decrease the number of cancerous cells. But in late stages, like stage 3 or 4, usually it works. I mean, it activates, it works, but it also helps the tumors to survive. I mean, this survival mechanism that's happening by autophagy always happen, but it depends on the age, stage.
Yeah, absolutely.
It's really
let me be more specific about how the autophagy or activation can just be detrimental for single cells. About the normals, we are talking about the normal cells. Can you make any, any underlying mechanism, underlying molecular pathway which are going to just face the supposed cells or face them, how can I say, dictate them to just go the cellular death?
Yeah, sure. Actually, there are some big differences between cancer cells and normal cells. In autophagy, we usually have low glucose in our bloodstream and in our body because it usually happens after long times of fasting. It means that we have to be under calorie restriction to activate the autophagy. When it activates, usually we need some energy sources in our body. So the body usually goes after having, after lackage of glucose or any carbohydrates, it goes to lipids. And if it consumes lipids, the ketone bodies appear here. So this is another phase that...
Pardon, sorry, I'm just talking about just single cells, normal cells. For example, as we mentioned by your classmates and your friends, and even you highlighted, the adapted autophagy is good for cell. Just for what? Just get rid of some insulting condition. But excessive activity is not a good. For what is not good? We are just talking about the single cells.
For what is not good? Actually, acidosis ketosis happens.
Okay, it's some normal condition just going to activate and stimulate autophagy after all. It's the autophagy activation followed by bioenergetic express. But inside a cell, where we have a excessive autophagic response, in that case, we have some overaccumulation of autophagosomes. So the lysosome could not afford the autophagosome flux. So we have an accumulation of autophagosome inside the cells, and we have some misfunctional protein inside that autophagosome. It makes additional stress to the cell. So it is not a good for cell, and cells should just get rid of these autophagosome as soon as possible. So the excessive autophagic response is not good, as I know. And so even that, in some case of the cancer cells, we have a higher autophagic response. You are right. By activation, some different kinds of ATGs, like ATG5 and 7, the cancer cell can just make them resistant to some therapeutic regimes, and yeah, you are exactly right. But even that over acceleration of the autophagy in case of the cancer cell is not also good. But from our side is a good phenomenon. Thanks a lot.
Thanks.
Mrs. Bazdar, the cancer and even tumor cells are one of the targets for autophagy. And as your friends mentioned, autophagy have two effects on the tumor cells. And just we want to know a little more about the relationship between tumors and autophagy.
Yes, as you mentioned, the autophagy can affect on cancer cells. But the important point is that calorie restriction can affect on cancer and cancer therapy with different mechanisms. The first one is autophagy. Autophagy, as my friends mentions, is a natural process for clean up the damaged or old cells in the body. And the second mechanism is ketones or ketones body. When the body in the calorie restrictions start to storage the glucose and the sugar, but switch to burning the fats for prepare the energy for body. The healthy cells can use this field for metabolism. And
you mean that the free fatty acid as their energy source?
The healthy cells can't use this full, but the cancer cells can't use this fulls, and their metabolism have some problems. And the last mechanism is the decrease the inflammation in the tumor and the microenvironment of the tumor. And many clinical research show that the calorie restriction in the cancer patients can affect the cancer therapy and a different method of cancer therapy, such as chemotherapy.
Great.
Yeah.
But if I'm going to make some brief conclusion about your remarks, in that case, from your viewpoint, the autophagy can just help our body to just get rid of the tumors, even to prevent the development of cancers. In one hand, the autophagy, or activated by calorie restriction, can just eliminate some of the age cells, which are sensitive to some gene mutation. So the rate of the cancer development will be reduced after a while. And even that, the production, this metabolic switching from the carbohydrate to just lipolysis is going to just increase the free fatty acids. The cancer cells could not use the free fatty acid because they are devoid, most of them are devoid of the mitochondrial activity, or even that they have some small number of the mitochondria in their cytosol. So they could not just appropriately use the free fatty acid for energy sources and even anabolic purposes. And the last one is the activity of immune cells, as I understand. So the calorie restriction is going to activate and stimulate the function of the immune cells and even their recruitment to the tumor sites. Thanks a lot. Mrs. Shapkizan, one of the key factors that can initiate the autophagy response is the initiation of some pathological condition and direct injury to the subcellular compartments like organelles. So do you think that this can be true or not? If so, then can you make a brief communication and brief comments about it?
Yes, Professor. One of the important process that is affected on calorie restriction and fasting is mitophagy. And what is mitophagy? As we know, we have millions of organelles in our cells that called mitochondria. And these mitochondria are very important to energy producing. Actually, they called power plant in our cells, and they are so important for producing energy for cells and consequently for our body. And as we know, damaged mitochondria is very dangerous for our cells because they produce lots of toxic sparks like reactive oxygen species that are very dangerous, and they injure the DNA and proteins and consequently cells. And on the other hand, damaged mitochondria are like, like a broken car that is stuck fully on speed, and they work non-stop. And when we are fast and when we are in calorie restriction situation, we give them an opportunity to break down and regenerate themselves. And
you're talking about the mitochondria.
Yeah.
Right now. Yeah. Great.
And activates properly and produce energy for anabolic purposes.
Yes.
And there is a crosstalk between mitophagy and autophagy, and mitophagy can activate autophagy and can affect it in the next step.
Okay, great. As I know, there are some effector, kinds of effector, which are translocate to the outer or external surface of the mitochondrial membrane. They are like a pink one and optineurin. These the activated factors. I mean, these factors can just provoke the common autophagy molecules like LC3. So after mitochondrial injury, we have activated autophagic flux. I mean that the numerous autophagism inside the cells, and so the cell wants to just eliminate the injured mitochondrial bodies.
When mitophagy starts, and it gives opportunity to cells to can uptake other
Healthy
mitochondria from the other cells, other healthy cells, by means of sending signal to other cells and in other cells in the environment.
Okay, great. The stress cells can just produce different cytokines, and even that some messenger, they can just alter the neighboring cell. They can just donate, I mean that the cells itself donate their mitochondria to the injured cell. This is, true. And the different documents have proven this phenomenon, which are normally happen inside our body under condition. So it's also possible that the injured cell or a stressful cell can just uptake the other organelles rather than the mitochondria. I mean that the Golgi apparatus, other subcellular compartments, which you can imagine. Thanks a lot. Mrs. Mamagani, I think that with plethoric effects of the autophagia, it can just affect the function of other organelles and tissue. Even that, I don't know, the activation of the autophagy in calorie-restricted individuals. How do you think that? And do you have any data about it?
One of the most impressive effects of calorie restriction is improved heart health, and studies shows that people who practice calorie restriction have a good heart rate pattern and heart health. That heart rate patterns like similar to people who are 20 years younger than them.
Great, very interesting data.
And also, calorie restriction reduces inflammation in blood vessels and keeps arteries more flexible and increase the risk of the high blood pressure. And also, research shows that these mechanisms are very relatable to autophagy in cardiac cells.
Yeah, cardiac cells.
Cardiac
cells. Great. As I know, you're right. You're exactly right. The autophagy response can just make the endothelial cells, which are covered the luminal surface of vessels, resistance to different metabolic changes by just they help them to just get rid of some of the injured molecules and even that some of the compartments and even that. But how do you think about free fatty acids? Are they detrimental for endothelial cells or cardiovascular system cells or not?
I don't have any information about this.
But it is logical to hypothesize that even that activation of autophagy in these kinds of a cell be beneficial because it makes some chance to cell that get rid of some misfolded protein, even that some injured compartment, subcellular compartment. Thanks a lot. Great. Some articles have proven that the calorie restriction can postpone the aging or senile changes inside the cell. By which mechanism do you think it will be happen?
Well, this is very actually structured question, and it needs deep analysis and deep answers for this. But I'm going to make it very general. Mm-hmm. There's a specific genes called sirtuins, and the subcategory is in sirtuins 1, 3, and 6. These actually genes they are very important. They have very important role in control the our neuroplasticity and neural aging system in our brain.
Also, they can be just applied to other cell types as well.
And they are also can reduce the calorie
consumption.
Consumption and gathering in our, for example, extra layer under the skin fat also. by the my friends here, they mentioned that actually by inhibiting the mTOR signaling pathways, we can actually trigger this, and by this function, the inflammation actually cascade will be cut, and by this, it will directly affecting to the aging. And by this mechanism, this is the main mechanism. For example, by the map of genes, there's a study in the Nature Metabolism journal. They analyzed this more than 2,000 genes involved in this
analysis.
And they are also they're altered, actually significantly altered by after the 30 days fasting. Four main type of fasting and calorie restriction, 400 calorie restriction methods.
400 what?
By the calorie restriction methods.
Oh,
in the four type, and it's a bunch of information there, and they actually run this test in the mice, okay, and in the flights also they run this test in the flights in the little glass by actually running this calorie restriction window actually feeding or time actually window feeding. When, for example, they by shaking calorie restriction groups, they've seen that the mobility or their movement will actually increase, and the other group that every time they have access to the food and the heavy calorie actually diets, their movements they are very significantly slow.
Okay, great. You mean that just by the calorie restriction, we have some improved movement, and it should be in some part to I don't know, postponing the scenario changes, as we know,
yes.
because the flies have some very limited lifespan, and so it should be
yes.
somehow true. Yeah, great. Thanks a lot.
Yes. And can I continue and
yes, absolutely.
conclude
absolutely.
scientifically here? After all of these concepts, my friends and I introduced and actually introduced here very deeply, the exact point is the calorie restriction is directly linked to the longevity. All of these mechanisms is for longevity. And what I mentioned, the movement, it is because of that. If some live organisms can move very fast, they can live actually very long. There are direct actually relationship between our aging process and aging and our legs muscle also.
Oh, great. Well, it's somehow true, but
and it's related to calorie restriction.
Yeah, absolutely. Absolutely.
Because of that, I mentioned.
Yeah, absolutely. By restricting some, nutrients, we educate the cells to just, mimic some behavior under circumstances. Is good. Even that excessive energy consumption can just produce the higher levels of the reactive oxygen species, which is detrimental, and just directed to just make some harmful condition to to subcellular compartment. Is it true? Yeah, actually. And the most important factors which are related to the senile changes are these free radicals, and so we should just restrict their production. But even we should not just deny and neglect their critical roles in some biochemical reaction. These free radicals are essential and required for some kinds of the anabolic and even proteolytic and even that some metabolic switching and even that reaction, and they should be just exist inside cells, but their levels should be controlled. I think that by my own idea, from my viewpoint, is that the calorie restriction can just make a good barrier and a suitable barrier to control their production inside the cell. This is my philosophy. Thanks a lot.